During alternative processing, single pre-mRNA can be processed in different ways and can produce alternative types of mRNA which actually give rise to different proteins. So what is the result? The same type DNA sequence give rise to different proteins. You can see that in this way, alternative processing actually complicate further our definition of genes when we define genes as a sequence of nucleotide that specifies the amino acid sequence. Because of alternative processing, the same DNA sequence can actually give rise to different proteins. So how this can happen, how alternative processing can happen? It is either via alternative splicing or multiple 3' cleavage sites. Here we have an example of alternative splicing. Here is an example of DNA which contains 3 exons, exon 1, 2, and 3, and 2 introns. All these exons and introns are transcribed into pre-mRNA. This pre-mRNA can be processed differently. There can be 2 alternative ways how it can be spliced. In first case, shown here, both introns, intron 1 and intron 2, are removed and exon 1, 2, and 3 are joined together. Alternatively, what can happen is during splicing, the 5' splicing site is used from intron 1, but 3' splicing site is used from intron 2. So not only 2 introns are removed, but also exon 2 is removed with them. And mature mRNA will have in this case only exon 1 and exon 3. So these 2 mature mRNA have different lengths and obviously will translate into different proteins. Another type of alternative processing is multiple 3' cleavage site. I want to make sure that you understand difference between 3' cleavage site and 3' splicing site. 3' splicing site you find within each introns. 3' cleavage site, it's at the end of the coding sequence, meaning at the end of the last exon of the gene. Now, look at this example. We have here exon 1, exon 2, which are separated by intron. Exon 2 basically can have 2 different cleavage sites, either cleavage site 1 or cleavage site 2. If the cleavage site 1 is used, we will obtain RNA which is shorter than if cleavage site 2 is used. So 2 different 3' cleavage sites can yield 2 mRNA with different lengths. Whether these 2 different mRNA will produce different protein will depend whether the position of the site is before or after the termination codon. Both alternative splicing and multiple 3' cleavage sites can be found in the same pre-mRNA transcript, and this pre-mRNA can be processed differently in different cells. Here is one example. It is pre-mRNA encoded by the gene for calcitonin. This pre-mRNA undergoes different splicing and different 3' cleavage in thyroid cell and brain cells. You can see this pre-mRNA contains 6 exons and 5 introns. In thyroid cells, 3' cleavage site at the end of exon 4 is recognized. So pre-mRNA is cleaved by the exon 4, introns are removed, and the final mRNA contains exon 1, 2, 3, and exon 4. This mRNA encodes for calcitonin, the protein which regulates calcium level in the blood. in the circulation. Contrary to that, in brain, a 3' cleavage site at the end of exon 6 is recognized. So the pre-mRNA is cleaved at the end of exon 6. However, there is also alternative splicing. By removing introns, also exon 4 is removed, and final mRNA contains exon 1, 2, 3, 5, and 6. And this mRNA gives rise to calcitonin gene-related peptide, or CGRP, which is involved in transmission of pain and can also regulate contraction of the blood vessels. So how is it possible that one pre-mRNA, one gene can give rise to such a different proteins? You remember spliceosomes. Spliceosomes are very complex complex structure which contain almost 300 proteins, and each cell can actually have different gene expressions. So different proteins might be included in spliceosome, and those different proteins would recognize different cleavage sites and might also mediate different or alternative splicing of the pre-mRNA. In addition to alternative processing, mRNA can undergo RNA editing. What is the difference? During RNA processing, pre-mRNA can be spliced or cleaved in different ways. However, sequences in the mRNA are complementary to DNA. So those sequences in mRNA could be found in coding strand of DNA. RNA editing, contrary to that, introduces new change sequences. RNA editing takes place after processing of pre-mRNA, and it introduces new sequence, change sequence, which would code for different amino acid. How it can be done? There are two major mechanisms which can mediate RNA editing. It can be via guide RNA, which contains sequences which are partially complementary to segments of RNA, or it can be altered enzymatically. For example, deamination. Deamination can trigger base conversion. For example, if you have cytosine, do you remember cytosine has this amino group? If there is deamination taking place and this amino group is removed, so cytosine is converted into uracil. So what would happen, for example, if you have codon for glutamine, CAA, and you deaminate that cytosine and convert it to uracil, you will introduce stop codon. So let's now look at how RNA editing carried out by guide RNA works. So guide RNA have sequences which are partially complementary to mRNA. So here is pre-edited mRNA, and here you have guide RNA. So this guide RNA has only partial complementarity to mRNA through which it anneals to it. This trigger cleavage in mRNA and basically adding or deleting sequencing depending on the sequence of guide RNA. In this particular case, you can see here there are new nucleotide added based on the sequence on guide RNA. So guide RNA adds or deletes nucleotides to the pre-mRNA and yields the sequences which were not encoded in the DNA. So let's now summarize what we learned up to now in chapter 13 and 14. RNA transcription is first step in gene expression, and it starts at the DNA promoter. It's a region of the transcription unit which is not transcribed in pre-mRNA, but it's critical for transcription because it's a place where transcription apparatus assembles. What transcribes into pre-mRNAs are exons, introns, and long 3' end. Now, once a pre-mRNA is transcribed, it is modified. First modification is adding of 5' cap. These 5' caps play a role in the interaction of mRNA with ribosomes and also increases stability of mRNA. This 5' end modification is followed by 3' cleavage. The cleavage site at the 3' end, it's downstream of consensus sequence, and it's followed by polyadenylation. So adding this poly A tail increases stability of mRNA and also play a role in the association of mRNA with ribosomes. After these two modification, pre-mRNA is spliced. What does it mean? Non-coding sequences, introns are removed and exons are connected. So mature mRNA contains protein coding region consisting of exons, 5' UTR, which is capped, and 3' UTR, which is followed by poly A tail. In these last slides of this segment, I would like to show you representation of the nucleotide sequence of the gene for human interleukin-2. IL-2 is a cytokine produced by activated T lymphocytes, which actually is important for their survival and proliferation. So it plays very important role in immune responses. So the simple gene is outlined here, and what you can see here, you can define the promoter. You see in promoter, you can see Tata box, TBP binding regions. Afterwards, you can see transcription start site, exons, and introns. You can also define within this sequence start codon and stop codon. So basically this is coding sequence here, and you can also find consensus sequence, and you know that downstream of this consensus sequence we will have 3' cleavage site. This is just an example of the gene, how you can actually define the sequence of certain gene, how you can define where it starts. You can look for Tata box, where it ends you are looking for stop codon and consensus sequence.